I've been in Houston many times. I am not a supporter of the Washington Nationals. So I feel okay to be here. When Diana asked me to give this talk, and I looked to see what the issues were, I probably went with the slide that Suzanne was talking about from reverse angle, because I'm starting a new company. So I said, okay, I'll take that and go backwards. And then also, there were going to be specialist speakers, so how would I talk about this general topic? So let me see if there. So I thought I'd tell a little bit about myself besides that's relevant to what's here today. So I am an academic that actually had a drug on the market, so I'm not only an ivory tower guy. I was the developer of a drug product called Maxide that at one time was in the top 200 selling drugs in the US. It was a reformulation of hydrochlorothiazide and triamterene, and it was sold by Letterly Laboratories. I directed the phase 1 and phase 2 studies for metformin before the drug was bought by Bristol Myers Squibb. So if you go back and look at the papers, I'm the senior author on the early metformin clinical studies. I've done 5 startups. One of them still exists, Urel Corporation. That's a liver on a chip company. And I've just started this new one, Squall Pharma. I was on the FDA Science Board, the first FDA Science Board. And I was on the Generic Drugs Advisory Committee, and I've been in the Institute of Medicine, National Academy of Medicine for many years and on all their forums. And at one time or another, I was a consultant for every drug company because I'm very old, so I've been around for a long time. So I'm actually going to just do one thing. I'm starting from the back. You're forming a new company and you need to get funding. Okay, that's not what this meeting is about, and I'm only just going to show you the slide. And the reason I'm putting it there is because that's what I'm doing right now. I'm trying to get funding for my company, and so these are the kinds of things that you need to do. What's the problem? What's the opportunity that you're addressing? What's your solution? Where's your data that supports what you're trying to do? What's your IP? What about regulatory? How are you going to deal with regulatory? And do you need to worry about reimbursement from your product in terms of whether the insurance companies are going to pay for it or not? How do you go to market? And what's your competition? What are your financials? And really, if you're trying to get investors, what's your exit strategy? That's what investors want to know. They're going to get their money in, and how are they going to get it out? And how you're going to partner, and what's your management team? So it's there, and I put it in my abstract. I'm not going to talk any more about it. This is a very old slide from the FDA, which I wish they still had. You can't get this slide, but it's a nice depiction of what goes on. Your accelerated and treatment IND, parallel track, that's old stuff. We now have fast track and a number of other things, but it nicely shows what you need to do. All this preclinical stuff that then leads to phase 1, phase 2, and phase 3 studies. And we're back here in the early stage, in the preclinical. What are we going to do with our compound, synthesized purification, the animal testing, and the proposals for the human testing? That's where we are today. So I've took it from the perspective of an IND, because what you need to do is do your IND-enabling studies, which is the second to last talk before you form the company. But you have to be able to do your IND studies, and that's where all of what we're talking about today really is the pharmacokinetics, pharmacodynamics, and ADME fits in, but it's a total broad picture. And you need to look at what that picture is. So IND-enabling pathway, this is my game plan for Squall Pharma that I've written up and I'm going to pursue. So I got to worry about synthesis, formulation, one of the topics for today, characterization and scale up, not necessarily things that we think about in terms of ADME. Stability, big issue we don't often think about. Preclinical verification in our animal models. Okay, you've got your animal model, that proved your drug worked, But now you have to carry out the animal studies that you're going to put in your IND, and can you verify that characteristics? The safety toxicology studies we're going to get into today. The additional patent filings that are going to really be based on the data that you generate in doing these IND studies. Your clinical development plan, your pre-IND meeting, and you file your IND with the FDA. So again, that's a list in my abstract. Those are the kinds of things that you need to pay attention to. We're concentrating in some of the intermediate steps today. So let's just go back to an IND, and I'm not going to go the actual everything that's in the IND, but these are the things that you need to pay attention to before even you carry out your ADME studies to make sure your molecule does meet the characteristics that are going to allow you to say that these studies are useful and will be accepted by the regulatory agencies as proving what you want to prove for your IND-enabling studies. So you've got to give them the name of the drug, the chemical name, the structure of the drug, the components of the drug, the quantitative composition of the drug, getting back to the analytical. If you're getting this from someone else, what about the synthesis, method of manufacture, where does it come from, and how have you qualified it? Because everything you're doing is a new drug. Okay? The old FDA definition of the new drug, it's been around for years. Any drug that is not generally recognized as safe and effective under conditions prescribed, recommended, or suggested in the labeling, that's a new drug. A drug may be considered new because of its composition of matter, new chemical, its use, its dosage, or its dosage form. It's readily recognized that an NCE would be considered a new drug. That was metformin. It wasn't ever out on the market before, and we carried out the clinical studies. However, a drug can be new resulting from composition of inactive ingredients. My first company, AVMAX, I had the patents on increasing bioavailability using GRAS substances to inhibit transporters and enzymes in the intestine. That's what AVMAX was based on. The proportion of the new drug can be, you change the proportion of the active ingredients. That was Maxide. Diazide was the biggest selling drug in the world at that time in terms of how much drug was sold. Maxide was a better formulation of triamterene and hydrochlorothiazide. And combination of inactive ingredients, or active or inactive, that's what we're trying to do with Squall Pharma. So, is the drug substance pure? Is your analytical methodology and potential contaminants adequate? Have you validated your analytical methodology? Quality control versus quality assessment. And what are your controls on the chemical process development? Have you selected, or do you select, the right physicochemical form of the drug substance? Process involves discovering, identifying, characterization, all available candidates of the API, polymorphic forms, solvates, hydrates, salts, complexes, co-crystals, selecting the best crystal form from the candidates, and determine the process boundary conditions among the selective API and other related forms by thermodynamic principles. A number of developments and new drugs haven't done this adequately. They've carried out their studies with really not a very well characterized form of that molecule. And they've got good data. And they've got animal data that shows that it works. But they really haven't characterized their drug substance. If you haven't characterized your drug substance, when you go to do your IND-enabling studies, you haven't met what needs to be the kinds of things that the regulatory agencies are going to look at. All the things that you have to consider in synthesis: fully characterizing the substance, the availability of the raw materials. Is your synthesis scalable? A big problem for small companies most of the time. Sure, I can make the chemical and I can make it in the lab, but you have to scale it up to be able to get the kinds of quantities that you need for your IND-enabling studies. The impurity profile. Do you have sufficient quantities for analytical development? The final salt form and crystallization defined, pH and organic solvent solubility, and reasonable cost of goods possible. All of these are scientific except the last one, but that's a cost issue. And in your analytical development and all the things you do, you've got to have this kind of information. You want to run force degradation, aids in the development of the analytical methods and especially specificity. Pre-formulation and excipient compatibility, aids in formulation approach and formulation possibilities. Open dish stability, aids in the selection of packaging and selection of expiry or retest dates. Now, I think we're getting into this, a little of this. Cindy, I think we'll talk about some of these aspects, but it's not something we usually think about when we go and do our pharmacokinetics and pharmacodynamics early in the drug development process. Because this is not getting a paper out. This is getting a drug approved and on the market to actually solve a clinical problem. Are the non-drug excipients pure? Again, you don't make your tablet or your capsule or your formulation only with the active species. You've got other substances in there. The CIRSE, Center for Regulatory Science around the country, is funding a bunch of studies now to look at the activity of the non-drug excipients. Because non-drug excipients can have activity. They can inhibit enzymes. They can inhibit transporters. They can have an effect on dissolution. Other non-clinical components, stability, the effects of time, self-life for the drug, excipients, formulation, the effect of temperature and humidity. Is your standard operating procedures, are they adequate? Have you done it correctly? And have you followed good manufacturing practices? So the definition of good manufacturing process, the requirement that drugs and the methods used and or the facilities of controlled use in their manufacturing process, packaging or holding conform with those practices will assure that such drugs meet the requirements of the FDA Act as to safety and have the identity, strength, quality, and purity characteristics that they purport or are represented to possess. If they do not, your compound is adulterated. So good manufacturing practices is not defined in the act or the regulation, but they are what the agency looks and it's are you meeting the criteria that are present today for having a quality form? Okay, so that's real quick background of the kinds of stuff that now allows you to get to your IND. Okay, and the studies that you're going to use for IND. So then, in that IND statement covering all the information available derived from preclinical investigations, that's what we're going to talk about today, and the clinical studies, and what about hazards and other aspects of what's out there. So I've taken all this from the IND, what's listed in the IND, the kinds of things you need to present in your IND. Okay, that was the background. Now you've got your chemical. Okay, so as I said, I'm old. So 30 years ago, I made this slide. It was the history of how we do with new chemical entities. So when I was a graduate student, Abraham Lincoln was my classmate. So when I was a graduate student, basically medicinal chemists in those days, or chemists, made about 500 compounds. And of those 500 compounds, about 10% of them they took and we ran studies of pharmacokinetics and toxicology in animal species. And of those 50, about 3 got into clinical studies. And in those days, about 1 drug got approved. So for every 500 molecules, we got about 1 drug approved. And these molecules were just based on what the chemists thought was interesting. So the chemists were making compounds, and then they happened to run to see if they were active, and they ran them through a bunch of screens. Then we got a lot smarter. We said, okay, we're not going to do that anymore. We're actually going to do rational drug design. In other words, look at what the receptor is, look at what activity we're going to do, and then we could make 5,000 compounds, And again, about 10% of them we would run through pharmacokinetics, metabolism, toxicology. 3 of them would get into the clinical studies, and 1 get approved. Okay? So we didn't change the number of approved, even though we were more clever in approaching it. And then we got this idea is, why do we even need chemists? Let's do combinatorial chemistry and high throughput screening. Every pharmaceutical company in the world did this. They said, okay, we're now going to take these tea bags and we're going to run compounds through them, we're going to make these compounds, and then we're going to put them into animals, and we don't need chemists anymore. We just do this, and we'll make now 5 million compounds. We can't do 10% anymore, we'll do 5,000, but they did that. 3 of them went into clinical studies, and not even 1 got approved. Why? Because actually you need to think about your drug development. You really need to go back to rational drug design. So although we spent billions of dollars doing all this combinatorial chemistry, really nothing came out of it except for saying, okay, I got a drug that works, and this is the backbone, and that's a clever backbone that a chemist has derived, and now I'll use combinatorial chemistry to modify that backbone. So we've really gone back to rational drug design, but we're even worse today in getting drug approval from the compounds that we look at. Okay, what are we talking about today? So what we need to be able to give to the regulatory agencies and to prove to ourselves are the pharmacokinetics and the toxicology of our new chemical entity. And what we're required to present is some acute studies, toxicity. Basically, it can be 2 weeks in a rodent and a non-rodent, in males and females. We start off with single dose, and then we dose rising studies. Long-term tox, 6 months at least, multiple dose rising. Now, what's really critical about these initial PK studies? What's really critical is the way the regulatory agencies work. You really cannot have a drug in humans that reaches concentrations that you haven't studied in your toxicology. So this sets where you're going to be able to run your human studies. But you also need to know what happens to the compound. So at the same time that you're doing these toxicology studies, you have to run the pharmacokinetics and the metabolism of those compounds, again now in a rodent and a non-rodent, male and female, single dose, dose rising, multiple dose, dose rising. Frequently, we will see no toxicity in single dose, dose rising, but low doses on multiple dose will give us toxicity. Why? Because single dose, the pharmacokinetics of the drug, the concentrations are in the body, they fall off, you get rid of it, we don't have the potential for accumulation out in a potential toxic site. So you may see single dose, really no toxicity whatsoever, but multiple dose leads to toxicities because your site of toxicity is someplace else beside in the systemic circulation. And the other simple study we run early is mutagenicity. Okay, now there are other preclinical animal toxin PK studies, teratogenicity run in rabbits, and carcinogenicity, lifetime studies in rats, but you don't run those early. You don't run those until you get farther on that you say, okay, I got a compound that works and I know the pharmacokinetics and toxicology. I think now I'm ready to go into humans. And then while you're running your phase 1 human studies, you're going back and running some of these preclinical studies to show the kinds of issues that you need to characterize. So in that, we need to understand metabolism. We should be identifying all the metabolites in each of the preclinical species, in liver microsomes, in hepatocytes, whole animals, and we usually compare it with human liver microsomes and hepatocytes. And this is useful in lead compound selection in comparison with competitors' compounds. We're very concerned about saturable metabolism. Saturable metabolism, if you have a drug and you're developing it and you see saturable metabolism, it's probably better to go someplace else unless you're first in class. Because saturable metabolism means you can't predict what's going to happen to that drug as doses increase or as different patients look at the drug. You've got linear kinetics, you know what happens. So we'll usually see saturable metabolism, and that's a flag that says, I don't want to deal with that. We're willing to deal with saturable absorption because that's not a toxicology issue. That means you're going to have less of the drug absorbed. So we'll deal with that kind of saturation. But usually we don't want to deal with saturable metabolism. Concentrations will go up. A very strong signal, your compound induces liver enzymes, go to something else. You know, to be able to get around inducing liver enzymes with the potential of DILI, drug-induced liver injury, is just something that you want to stay away from. And eventually, you're going to have to run a radio-labeled or a cold-labeled mass balance study with the lead compound to make sure that you understand all the metabolites and you have mass balance. Now, I just consulted with a company that compound going to the FDA and probably will get approved that had very clean pharmacokinetics in rats, mice, monkeys, and humans, but toxicology in dogs. Okay, toxic substance in dogs. Should the company pursue a compound that has a toxicology, serious toxicology in one species, but not in the others? So what does this company do? What they have to do then is to go back and explain why the toxicology is different in the dogs than everyone else. And they were able to do this. A different metabolite was formed. The pharmacokinetics were very different. But the hoops that they had to go through to prove to the regulatory agencies that they weren't accumulating, and they could identify what, or they supposed what the active metabolite was that was causing the toxicity, and showed that it wasn't present in humans. And what did the agency want? I want to see a lower level of sensitivity in your assay. How do I know that your assay, you didn't measure at Cmax? Go back and measure at Cmax. They ran through all those hoops, and this drug is going to be approved. It's very close to being approved. If you don't know the mechanism, and I've actually published some papers with Takeda on Facilogam, a diabetes drug, that they have toxicity in dogs and they don't understand the mechanism. They drop the drug. Not understanding the mechanism, realistically, you're never going to be able to prove to the regulatory agency that you understand what happens with this drug. So this is a really critical component of your early pharmacokinetics drug metabolism. So now I want to go the kinds of things that I do and sort of useful, that could be useful to you in drug development. So in 2005, we made a very simple discovery based on the FDA biopharmaceutics classification system. And those of you with a pharmacy or pharmacology, clinical pharmacology background will know what that is. Gordon Amidon and his colleagues at the FDA in 1995 published this paper that said, let's put drugs in 4 categories, very simple categories based on permeability and solubility. And at that time, the basis was if you had a class 1 drug, highly soluble, highly permeable, there is no evidence there's any problems with those drugs in terms of pharmacokinetics unless you make a poor dosage form. And therefore, once you've run all the studies, you don't need to run bioequivalent studies with all your dosage forms. If you've got a class 1 drug, you can just show that your dosage form releases the drug. So I sat through many talks looking at biopharmaceutics classification system, and here's the kinds of slides that would be shown. Here's the class 1 drugs, high solubility, high permeability. Class 2 drugs, low solubility, high permeability. Class 3 drugs, class 4 drugs, poor permeability. I looked at slides like this over and over again until one day I said, wait a minute, I know pharmacokinetics. I know that all those drugs that are class 1 and class 2 are metabolized in humans. And all those drugs that are in class 3 and class 4 are not metabolized in humans. The major route of elimination is excretion of unchanged drug in the urine or bile. And is this permeability characteristic setting off the difference between how humans handle this drug? And so basically, we published this paper in 2005 that said, if your drug has high permeability, in vitro high permeability, or intestinal high permeability, in humans, that drug's going to be metabolized. And if it's low permeability, in humans, that drug will not be metabolized. And it's amazing how well this works. It really does. And you've never run the studies in animals or humans, and you can predict in humans what the major route of elimination is going to be. Not what enzyme, but it's going to be enzymes. Not what transporter, but it's going to be transporters. So we said, let's talk about a different way to look at this. If you've got high permeability, the drug's going to be metabolized in humans. If you've got low permeability, it's not going to be metabolized in humans by its major route of elimination. And so we said, well, let's take the BCS and convert it into something which we called the BDDCS, Biopharmaceutics Drug Disposition Classification System, that just uses metabolism and solubility. And metabolism is very much related to permeability. So why should this happen? Why should drugs that are high intestinal permeability be metabolized and drugs that are low intestinal permeability be not metabolized? That seems like a really strange thing. We're looking at a kinetics in the gut to predict what's going to happen in humans. And so it took us some time to figure out what was happening. So we suspect that what happens with the high permeability rate, let's take a drug like diazepam, has a very low hepatic clearance, yet none of it's excreted unchanged in the urine. So why is that? Well, certainly the drug gets into the urine, but it's high permeability. So what happens in the urine as you do your glomerular filtration, 120 mils per minute, but your urine flow is only 1 mil a minute? Basically, fluids are reabsorbed. Okay. So what happens? The concentration in the kidney tubule gets higher. And this drug, if it's high permeability, there's a concentration gradient, so it goes right back into the blood. So the only way the body can get rid of that compound is to change its permeability. And how does it do that? It metabolizes the drug. And so that's why high permeability compounds are metabolized. What about low permeability compounds? Well, they can't get reabsorbed because they're low permeability, or they need a transporter to get permeability. So you excrete them in urine or you excrete them in bile. But there was more to the discovery. We could actually take some understanding of the importance of transporters. And what we said was transporter effects for class 1 drugs are going to be minimal in the gut and the liver and clinically insignificant. Even if the drug in vitro shows it's a substrate for a transporter, we can't find clinical data that says that's important. But class 2 compounds, also excreted by metabolism, but they're low permeability, they're low solubility compounds. You're not saturating transporters, even though you have a high passive permeability, you don't have drug going by, you are going to have the potential for transporters. Class 3 and class 4 compounds, of course transporters are going to be important because they can't get by the membranes without the transporters, both in the gut and the liver and the kidney. So what are the implications for a new molecular entity or a drug-drug interaction? Well, for an NME, measuring a surrogate of human intestinal absorption, such as CACO2 permeability or even PAMPA, and we've published these studies, allows prediction of the major route of elimination in human prior to dosing either to animals or man. And furthermore, one knows what drug-drug interactions relating to metabolism are potentially going to be important or not important. The major advantage of BDDCS is the prediction of drug disposition and drug interactions can be made before any animal or human studies are carried out with the new molecular entity using just an in vitro measure of membrane permeability. Now, what the FDA does for solubility, they say take the highest dose strength of the drug that's approved Is it soluble in 250 mils of water? If it is, then it's high solubility. But we're back real early. We don't know what the dose is going to be in humans, and we want to be able to make this prediction. So faculty at State University of New York at Buffalo basically went and looked at all our data. We published this huge data set, and they said, all you need to do is look at a 0.3 mils per minute cutoff. So if your drug over these entire pH range is soluble at 0.3 mils per minute, that's going to be a high solubility drug. If it's not soluble, it's going to be a low solubility drug. So this is the ability to make the prediction very early before you've ever taken that drug and put it either into animals or humans. And there's a lot of things that we've published that we believe BDDCS can be useful for. Potential drug-drug interactions, whether transporters and enzymes are going to be important, pharmacogenomics, brain disposition, food effects. I'm going to talk about a couple of these. So potential DDIs. Class 1 drug, it's only going to be metabolic. Your interactions are not going to be transporter, even though your in vitro studies showed that that drug was a substrate for transporters. Class 2 drug, extremely complicated. Major route of elimination is metabolism, but transporters can be very important. In the gut, it appears to be only efflux transporters, in the liver, both uptake and efflux transporters. Class 3 and 4 drugs, it's going to be transporters that you need to look at in terms of your DDIs. So what about food effects? Every drug molecule that goes to approval, you have to say, what's the effect of a high-fat meal, and present that to the regulatory agencies. So back in 1999, when there was only BCS, the late David Fleischer and colleagues, Aziz Karim at Cyril in those days, actually looked and said, well, if you look at class 1 drugs, and let's just look at F extent, how much gets absorbed. It appears that class 1 drugs, high-fat meals don't have any effect on them. Class 2 drugs, most of the time, you increase bioavailability with a high-fat meal. Class 3 drugs, most of the time, you decrease bioavailability with a high-fat meal. Class 4 drugs, there's not enough of them, and there's not good data to make any guess whatsoever. We looked at that and we said, that's exactly what we would expect if high-fat meals affect transporters. Because transporters in the gut, the transporters in the gut that are important are the efflux transporters, PGP, BCRP. Those are the drugs that push the drug back into the intestine. If you inhibit those transporters, you're going to increase bioavailability. So maybe what high-fat meal does for class 2 drugs is increase the bioavailability by inhibiting transport. Class 3 drugs need a transporter to get in. So if you inhibit the transporter, if you inhibit the transporter, you're going to decrease the bioavailability. So Joseph Custodio, who was in my lab at that time, said, well, let's go look and see what's in high-fat meals. Does anything in high-fat meals predict what's going to happen with transporters? And what he found was that monoglycerides, at the high concentrations you get with high-fat meals, affect transporters, but it's only right 70% of the time. So 70% of the time, you'll be able to predict before you ever dose this drug in humans what a high-fat meal is going to do, but 70% isn't good enough for the regulatory agencies, so you still need to run the studies. But don't waste your time doing animal studies with food effect. BDDCS will predict better than animal studies, and you don't even need to run the studies, but you still got to run the human studies. And 70% of the time, you'll be right. And food effects and what happens in the intestine is a big quandary. We don't know what goes on. We know 70% of the time what goes on, but 30% we don't. Okay, what about getting into the brain? What we say is that class 1 drugs are not affected by transporters in vivo. So if that's true, what we usually think the blood-brain barrier are transporters, P-glycoprotein, and other efflux transporters. If the drug is a substrate for those transporters, it's going to be protected from getting into the brain. But class 1 drugs we predict are clinically not significant in terms of their transporter effects. So we say, and have shown in these papers, that really all class 1 drugs get into the brain. So if you don't want a drug to get into the brain, you don't want a high solubility, high permeability compound, because it will get into the brain no matter what. So normally we think we want a high solubility, high permeability compound, but maybe not if we want to keep it out of the brain, because class 1 drugs will get in. So let's talk about drug-induced liver injury. So recently we published 3 papers in the last 3 years concerning hypothesized predictive measures for drug-induced liver injury. And what we did was go back and look at all the in vitro measures that people use to predict DILI. And we did this in conjunction with National Center for Toxicology Research at the FDA, using their database. And basically what we showed was, if you look at the drugs in terms of their drug label for approved drugs, if you look at, there's no mention of liver toxicity, or if it's discontinued and withdrawn, look what's happening. For the drugs that have no DILI potential, yellow is a class 1 drug, green is a class 3 drug, high solubility drugs. Red is the class 2 drug. As you go for more and more toxicity, you see class 2 drugs basically becoming the drugs that cause DILI, class 1 and class 2 drugs not causing DILI. And if you look at the DILI data from the FDA data set, you see the same thing. Severe DILI, class 2 drugs. So basically, the toxicity risk of class 2 drugs much more than class 1 and class 3 drugs. You're certainly not going to say, if I have a class 2 drug, I'm not going to develop it, because about 30% of drugs on the market, we wouldn't have cyclosporin, we wouldn't have tacrolimus, we wouldn't have all the statins if we didn't have class 2 drugs. Okay. But those are the drugs that are going to have potential for cause DILI much more than other drugs that are class 1 or class 3 drugs. So pharmacokinetics, and Johnny's going to get into this, you know, what's the bioavailability? Is clearance too high? Is half-life too short? Is clearance too low? Is half-life too long? Can you scale the animals to man? What about the nonlinearities? An important characteristic, you should never make any decision about whether a drug is useful or not based on its protein binding. That's a complete fallacy. It doesn't make any difference whether it's high protein bound or low protein bound in terms of whether it's going to be active or not active. The only thing that can affect that is cost of goods, because if you've got a high protein bound drug, you're going to have more drug in your dosage to get the concentrations that are going to work. So I'm getting, how am I doing? Do I have my time? Okay, I still got 5 minutes. Okay, so back in 2011, Pharma put together a group, all their companies, major companies, and said, let's go look and see how well we do in IVIVE, predicting from in vitro measures what's going to happen in vivo. Okay, and so they looked at all these drugs, and they published these papers in 2011. And Malcolm Rowland and I were sort of the overviewers of this information, and we wrote the lead commentary. We didn't do any of the work. That's normally, I never do any of the work, I just take credit for it. But here's their summary. Although some prediction methods are better than others, there is currently no universal outstanding method to predict whether a drug, you're going to get the right kinetics of that drug. Prediction of human PK after IV dosing is much better than after oral administration, but most of the time we want oral dosing, and that's what we want to predict. Even the best methods could only predict within a factor of 2 for 45% of the drugs that the pharma group looked at. And if the class 1 drugs, high solubility, high permeability, they were best, but never better than 45%. And no one clinical species was found to be superior to others, nor were multiple species data shown to be superior to data from one species. Now, we generally believe monkeys are going to do a better job. But if you go back and look at all the literature, it's not really true that it does a better job. In some cases, it does. And you look over a bunch of animals and you'll see it. But if you go look at everything, monkeys don't always give you the best predictions. And PBPK on average is no better than allometry. I think Johnny's going to talk, or you're talking about allometry.
A little bit.
Yeah, allometry. Allometry just says, I'm going to take animal data and predict what happens in humans, making some crazy characteristic. PBPK is what the agencies love right now. Every company, you have to do PBPK. My problem with PBPK is you always need your fudge factor. You almost always need a fudge factor to make your prediction. So PBPK is really useful once you know what your drug does, and then you predict all the things that can happen in terms of liver failure or renal failure. But taking in vitro measures and scaling up to do PBPK from the beginning is a crapshoot. And basically what you do is you say, okay, I did all these numbers from in vitro, but I don't get the right answer, so what do I need to do? I change this, I change that, and basically any of you doing computer fitting, you know you can always fit the data. If you can change the parameters, you'll fit the data. So it's, in some sense, anti-scientific, PBPK. But it's still very useful once you know all the characteristics of your drug. But right from the beginning, it doesn't work. So I've listed some slides that you can take a look at in terms of safety testing and non-clinical safety testing. And I'm going to say, finally, one of the things that we've said in our DILI papers is we don't really do a very good job in predicting any of the in vitro measures, but maybe it's because what we're doing is only looking at hepatocytes or microsomes in a very short time frame. Maybe we should be looking at long-enduring hepatocytes. Now, there's a bunch of companies out there making hepatocytes that last for 21 days. And there's some very nice data, and I'm not going to talk about it here, but I have a conflict of interest. I'm a founder of one of these companies that basically shows that in 21-day studies or 14-day studies, I can do a terrific prediction of DILI versus looking very early. So in our analysis, none of the in vitro methods ever predicted you're not going to have DILI any better than just keeping the dose low. Keep the dose low, you'll have less prediction. But BDDCS actually is just as good as all of the in vitro DILI predictive mechanisms. You don't use BDDCS to say whether a drug's going to be toxic or not. You use BDDCS to say, if your method is no better than the predictions from BDDCS, just avoiding class 2 drugs, then you don't have a reliable method. So here we are back again. I've tried to give you some data from our lab relating to various aspects of the new development process.